Good evening, everyone. So tonight we're going to be talking about empowering CCM patients through research participation. And I have with me tonight on the panel, first off, I'm Connie Lee. I'm the president and CEO of Angioma Alliance. And tonight I'm joined by Spencer Rosero, who is here from the University of Rochester. He is a 2021 grad with a Bachelor of Science in neuroscience and a minor in economics, and I'm going to read his creds here. He is a clinical research coordinator at the University of Rochester Center for Health and Technologies, and he's assisting with the development of patient-reported outcome measures, primarily for neurological diseases. The outcome measures are going to be used in clinical trials and for clinical monitoring, and of course, the most important one he's working on right now is the CCM Health Index. I'm also joined by Dr. Barney King. Dr. King has extensive experience in drug development across a broad range of therapeutic areas, specializing in strategic product development and execution. He's board certified in internal medicine and cardiology. He has an MBA from the Wharton School. And for the past several years, he's been focused on the development of drugs for rare diseases. He's been working with Recursion Pharmaceuticals for the past 2 years. I've had the pleasure of working with him for at least the last year, and I'm glad he's joining us today. He'll be here mostly for the Q&A session. And also here is Adrienne Chalene, who is our clinical research coordinator, and she'll be talking just a little bit about the recruitment efforts that are going to be starting for the CCM Health Index. And then at the end, we're just going to keep rolling along here. Dr. Lisa Boyette is coming back. She was the presenter last month talking about Recursion's Rec 994 trial, the start of the phase 2 trial. She does have a little bit more of an announcement today, and she's going to share that with us at the end of our presentation before the Q&A. So I'm going to start first with a share screen. Hold on here. Let me find my slides. So as I mentioned, we're going to be talking about empowerment through research participation. So, I think all of us, when the diagnosis first comes to us of CCM, it feels big, it feels overwhelming. And at this point, we don't have a whole lot of answers. We're working on it and we're going to get there soon, but we don't have them yet. So I think it's easy to feel that there's nothing that can be done. It's easy to feel powerless and to feel helpless. And I can tell you that was a big part of my motivation for starting Angioma Alliance, is coming out of that space of feeling powerless. So what I learned by starting Angioma Alliance, but what I think all of us can learn, is that individually we can each make a difference. CCM is only going to be solved if we all participate. And at this point in time, although we do a lot of fundraisers here, really your time is just as valuable as your treasure. We know that the only way that treatments can happen that are effective is if treatment developers understand what the patient experience is and what our preferences are, what's important to us. That's what's critical to advancing treatments. And so when you share your experiences, you make a difference. You make a difference for the future of the illness in general, and possibly even for you personally, you and your family's experience with the illness. So, and honestly, the fact is that clinical research participants are medical heroes. You are. When you participate in a clinical research project, you are really participating in a heroic act. We are leading the way to a cure. And this is the bottom line message here, is that research participation creates empowerment. If you don't want to feel helpless or powerless anymore, just sign up for research that gives you purpose, and it's meaningful. So especially now, this is important because we are now at a point where the research has moved beyond the petri dishes and beyond the animals, and people are what's needed in order to keep research moving forward. We are the only ones who can provide those answers. There is no they out there. it's us. We're the ones that they need. They need our help also to develop the tools that are specific to CCM, like the CCM Health Index that you'll be hearing about today. And they will need our participation in treatment trials. And you know that if we're not participating in trials, they are not going to succeed, and we are not going to be able to find out what is helpful for the illness and get to a cure. So how would you want to start if you were wanting to become a participant, a medical hero, someone who is empowered and making a difference? Well, the first thing to do is to just go online from the comfort of your own home and join the Angioma Alliance Patient Registry. Everything starts from there. It is a simple process, and what we want you to know is that it is important. As I mentioned, every clinical researcher and treatment developer wants to know what our membership looks like. And some specific questions I get asked are, how many people have the familial form versus the sporadic form? Which mutations do they have, and how do they fall out into those mutations? How many people have had a hemorrhage, and how long ago was that hemorrhage? Who's having seizures? How many people are having seizures? How many have disabilities? How much of your population has an impairment that keeps them from working? How many people were diagnosed as children, or how many children are there currently diagnosed? And then how many people are in the US versus how many people in our database are international? Those are all questions that I have been asked that Adrian will go into the patient registry and try to get answered based on the information that you provide in your registry profiles. The other thing that we do is we specifically look for new registrants who qualify for some of the studies that are ongoing. So if you register and you had a hemorrhage in the last couple of months, we will reach out to you and say, have you heard about the CASH studies, the trial readiness and the atorvastatin trial? Because we want you to at least be aware of them. There's no obligation to participate, but you can't participate if you don't know that it exists. So that's our point, is in at least getting you that information. The CCM2 exon 2 through 10 deletion study, the Breaking Barriers Initiative, those are things that we are regularly looking for folks to be participating in, and there's countless others. So what do you get aside from that empowerment and the fact that you are driving research forward? Well, when you join the registry, you also get access to data, to the registry data itself, not individual data. You get access to the aggregated data that we collect. So there's an example here on the screen. How many cavernous angiomas does the affected individual currently have? That was part of our most recent survey. 1,781 people answered that question, and you can see that almost 900 of them had one. 450 about had 2 through 10, 250 had more than 10, a number had 0, and probably those are people who were in the registry post-surgically, and then there were a number of people, about 150, who just weren't sure what category they fit into. Oh, I'm sorry, I skipped ahead and I shouldn't have. The other thing, because there's so much more, you also get early notice of research findings. When a new paper is published that is relevant to our patients, we summarize it in lay language and we send a newsletter out to everybody in the registry first. Before it goes on Facebook, before it goes on our website, the folks in the registry get that information. The registry is also where you would request free genetic testing. Angioma Alliance has a program that offers free testing to anybody who has multiple cavernous malformations that can't be explained by a DVA or a history of brain radiation. This is where you would start to make that request to see if you could be part of that program. And then, as I mentioned, you get notified if you qualify for a study. So the next question is probably, can anybody join the registry? The answer probably is yes. So if you have ever had a CCM diagnosis or your family, your child has ever had a CCM diagnosis, you can join. The registry is international. We conform with GDPR standards. And if you're not in Europe, you don't need to know what GDPR is. but we do conform. It's international. We include both adults and kids. We include people with spinal lesions. We get asked that all the time. If you only have a spinal cavernous malformation, we want to know about you. You are so important to us. And we do include people who are post-surgical. So we want to know what disabilities you still have. What is your experience still post-surgically, even if you no longer have a lesion? We need that information. So how do you join? Angiomaregistry.org is how you join. Just go there. There's like every page on our website has a link to there. So just go to our regular website, angioma.org, and pretty much if you just press on anything, it's going to take you to the registry. It takes about 20 minutes. After you create your account, there will be a survey that you'll complete where you will share your experience of your history with CCM. And then if you have a little extra time at that moment, or if you could come back later, there's a second shorter survey that asks about the other illnesses that you either have had in the past or currently have, and we call those comorbidities. So what if your condition changes? You registered a few years ago and now all of a sudden something has changed. Well, every 3 months, we send you an email that gives you a link to a very short survey, 5 questions. And the questions are basically, have you had a hemorrhage? Have you had a new seizure? Have you had a headache? Have you had surgery? Have you had other symptoms? And if you answer yes to any of those, we'll reach back out to you and try to find out more of the story. It's really important, though, that you try to respond to those every 3 months, because we also want to know about people who aren't having more symptoms. And what if you change your mind? You are allowed to change your mind. If you decide that you no longer want to be part of our registry, you just contact us. Coordinator at angioma.org goes straight to Adrian, and we will make the arrangements to have your data removed from the patient registry. You will no longer get emails from the patient registry with updates or asking you to fill out more surveys. You can be done, and that's just fine. So what other research projects are out there? I mentioned the atorvastatin trial and the Trial Readiness Project. Those go hand in hand. The atorvastatin trial is happening just in Chicago. It's getting near its end of recruitment. So we've had a very successful 3 years of getting patients to them. And as most of you have heard Dr. Wad talk in numerous webinars, atorvastatin is a cholesterol lowering drug that we think may also be useful for stabilizing cavernous angiomas that have recently hemorrhaged. The Trial Readiness Project, I have a picture of a nest there for a reason, because the Trial Readiness Project reminds me of nesting, of what women do before they have babies. They're getting ready. They're getting ready for clinical trials. Among this group of institutions, they are standardizing the data they collect. They're trying to figure out how many patients they actually have and what they look like. They're standardizing their imaging. They're nesting, and they need your help to really do the nesting, to make sure that what they're creating there is going to be structurally sound. Finally, the Brain Vascular Malformations Consortium is now in its 13th year. It's a project that we were really important in getting started. It is only for people who have familial CCM, but it is now open to both adults and children. You can see the sites that are involved: the University of New Mexico, UCSF, Barrow, University of Chicago, Boston Children's, and Cincinnati Children's are all part of that. A lot of it is collecting just natural history data. You give a blood sample so that they can do some genetic testing and look for biomarkers. And there's really been a lot of papers that have come out of that already. It's sundowning now. It only can go for 15 years and then it will stop. So if you're going to get on the ride, you've got to do it now. So again, clinical research participants are medical heroes, and we would love to have you join our group of medical heroes. The next major recruitment that we're going to be doing is for the CCM Health Index, phase 4, I believe it is, of the CCM Health Index. And at this point, I want to turn it over to Spencer, who will tell us a little bit more about the CCM Health Index and what that is.
Great. Thank you so much, Connie.
You're welcome.
All right. Good evening, everyone. My name is Spencer Rosero. I have a background in neuroscience and work as a research coordinator for the University of Rochester Center for Health and Technology's Outcomes Division under Dr. Chad Heatwall, who is the principal investigator of this project. On behalf of our team, it is our pleasure to be here tonight with all of you. Thank you to both Connie and Adrian for the invitation to be on the panel. And thank you all listening today for your continued participation in driving the field of CCM therapeutics forward. Today, I want to focus in on a specific area of therapeutics, clinical outcomes assessments, otherwise known as COAs. COAs are a group of measurements designed to assess how a patient feels, functions, and survives. Currently, there are four main types of COAs: performance outcome measures, clinician-reported outcome measures, observer-reported outcome measures, and lastly, patient-reported outcome measures, also known as PROs or PROMs. Together, I want to discuss patient-reported outcome measures in the field of CCM. A PRO has been designated by the Food and Drug Administration as any report of the status of a patient's health condition that comes directly from the patient without any interpretation of the patient's response by a clinician or anyone else. So what exactly does that mean? A PRO is designed to specifically measure any subtle changes in a patient's health solely based upon their input. A PRO highlights the notion that no one understands a patient's experiences, struggles, and triumphs better than the patient themselves. While PROs are not the sole measure of therapeutic efficacy in clinical trials, they are a quintessential element of the process, permitting patients an authoritative voice, allowing their perspective to be heard, seen, and appreciated in the ever-changing world of therapeutics. They transform patients from the center of focus into an active participant at the therapeutics table of discussion. In addition, PROs have many other benefits as well, such as being able to be completed without external administration, ability to track small but clinically relevant changes in health, be an FDA-approved mode of drug labeling claims, and many others highlighted here. In the world of therapeutics for CCM, there are currently a couple outcome measures which are being discussed for use in therapeutic trials. These are the NeuroQoL, the PROMIS, and the EuroQoL. While each one has their advantages, their overall lack of disease specificity make them incomplete measurements to measure CCM disease burden. Let us discuss the NeuroQoL, also known as the measure of quality of life in neurological disorders. The NeuroQoL is a patient-reported outcome measure that assesses the health-related quality of life in both adults and children with neurological disorders. The instrument has been developed in depth with 17 different domains and subdomains based on patient input. However, an important fallacy of the NeuroQoL and its ability to be used in the CCM population is the lack of CCM disease-specific patient-reported data used in its creation. Throughout its creation, input was used from stroke, MS, ALS, PD, epilepsy, and MD patients, but not CCM patients. The lack of CCM disease-specific input places limitations on the NeuroQoL, as our group believes that CCM and every other neurological disorder holds unique and disease-specific challenges that cannot be captured through a generalized instrument. The next outcome measure currently used in CCM clinical trials is the PROMIS. PROMIS is short for Patient-Reported Outcomes Measurement Information System. This specific PRO is designed to measure physical, mental, and social health in both adults and children of the general population who suffer from chronic conditions. While as a generic PRO, this outcome measure has shined, its lack of disease specificity limits its overall sensitivity and efficacy in disease-specific therapeutic trials. Once again, you can see how this specific PRO has limitations in its depth of understanding and assessment of disease burden in patients with CCM due to its lack of insight into the unique obstacles that the CCM population has to overcome. The last outcome measure I want to touch upon tonight is the European Quality of Life Scale, or for short, the EuroQoL. The EuroQoL has been cited as a generic and versatile tool to measure quality of life across a variety of different patients and diseases. Like its counterparts previously discussed, the lack of disease-specific data from CCM patients and lack of specificity on CCM as a unique disease make this measurement an inexact measure of novel therapeutic effects in the CCM population. Our group believes that generic patient outcome measures hold a historically significant place in the field of developing therapeutic outcome measures and continue to provide value as an interim tool to bridge the gap between patients and therapeutic development teams. However, we strongly believe that their place in disease-specific therapies is limited, and their replacement with disease-specific outcome measures provide exponentially more profound insight. They give the voice of a patient the privilege it deserves to be heard on its own, to bring their unique experiences and viewpoints to the table, specific to both the patient themselves and the disease which has defined much of the experiences of their life. To combat these issues, over the past 2 years, our group has diligently dedicated itself to the task of creating an outcome measure specific to the CCM population. Our group is proud to announce the Cerebral Cavernous Malformations Health Index, or as we like to call it, the CCM High. The CCM High was developed through a multi-stage process. With each process, we identify an aspect of patient experiences and perspective, then with the utmost scrutiny, craft and hone it to be highly specific and descriptive. At first, we develop an understanding of the patient's perspective of the disease. Through qualitative interviews, we are able to identify both general and unique symptoms that patients with CCM experience and the impact that each has on their life. Continuing to develop the instrument, we took this information and transformed it into an all-encompassing survey to identify symptoms deemed most important to patients experiencing CCM. And at the completion of this phase, our group obtained the necessary prevalence and disease-specific impact information needed to draft our initial version of the CCM High. At Chet Outcomes, we hold the strong belief that something can always be improved, refined, and simplified. In accordance with this belief, in the next step of our development, we took the initial version of the CCM High and distributed it to a number of people with CCM. These participants gave in-depth feedback through one-on-one meetings with our PRO specialized research coordinators, which gave our group the necessary insight to further improve the CCM High. Specifically, patients illuminated items on the CCM High that were missed, redundant, common points of confusion. At the end of this process, our team was confident that only questions that were easily understood, of highest quality, and pertinent to the patient population were on the CCM High. With this feedback, we were able to refine our instrument further, clearing any misunderstandings and addressing any inadequacies it may have. With this most current version in hand, our group dove into one of the last phases of CCM High development. During this phase, the CCM High underwent several statistical and analytical tests to ensure the instrument was responsive to the small and gradual changes in patient CCM health, which are so crucial to the instrument's efficacy. In addition, our survey underwent test-retest reliability, in which participants took the CCM High at 0 days and then again at 2 weeks' time. This ensured that the instrument was both a responsive and an accurate measure. We are proud to say that at the completion of this phase, the CCM High was determined to be relevant, innovative, yet still easily usable CCM-specific patient-reported outcome measure. The CCM High is a CCM-specific outcome measure which encompasses 20 individual thematic components, each encompassing more specific symptoms within. The CCM High is specific, sensitive, relevant, and enjoyable to use. Not to mention it can be completed in 15 minutes or less. Our group is confident in the CCM High's ability to depict the unique CCM patient perspective each of you hold, and only you have the ability to tell. It gives each and every one of you the authoritative ability to describe your struggles and triumphs in a therapeutically relevant way, which can then be used in therapeutic development. We are excited to announce that this instrument is ready for use in any CCM therapeutic trial in need. Users of the CCM High overwhelmingly agreed that the CCM High obtained an accurate perspective on how they functioned from day to day and how they feel. One user even remarked, "This survey is a fine measure of the change in how I feel about how the seizures impact my life today versus how they did when they first started," highlighting the sensitivity and responsivity of the CCM High. So where do we go from here? In addition to putting the CCM High in the hands of therapeutic trial teams, we are excited to gain new insight into CCM disease progression over time. Although current literature out there exists on CCM, further understanding of disease progression from a patient's point of view will provide valuable insight as research in this field moves forward. This longitudinal study will determine the specific areas of symptomatic burden that progress the fastest and ascertain what demographic features are associated with a faster or slower progression of CCM. Also, we aim to optimize instrument responsiveness to further refine the CCM High. We anticipate that this data will allow for the subtle, clinically important differences to be understood, which allows future clinical trials to focus in more efficiently and focus on what is most important to patients. We are excited to announce that this study will be done with the help of both the Angioma Alliance and Recursion Pharmaceuticals. In the past, we have partnered with both and are very thankful for the relationship we have with each of these great teams. Similar to the past, Angioma Alliance will be handling all the recruitment logistics of the study, and any interest and participation should be directed towards their team. I want to take a moment to extend a huge thanks to the rest of my team at Chet Outcomes Division for all their hard work and dedication to this project. In addition, I want to take the time to thank Dr. Connie Lee and Adrian Shailene and the rest of the Angioma Alliance team that makes all our work possible. I also want to thank the team at Recursion Pharmaceuticals for their continued partnership in our research. And last but certainly not least, I want to thank all of you listening today for your participation in this field of research. As a group, you gave us the privilege of helping give you the individual patient voice that you all deserve. The CCM High could not have been created without you. I invite you to join us as we continue forward with our new study, and we'll be happy to answer any questions at the end of the webinar. Thanks. Pass it back to Connie.
Thank you, Spencer. That was great and really informative. We are so excited about being able to enter this next phase, and a lot is going to be asked of you, and Adrian is here in order to share what it is that's going to be asked of you. She just turned her video back on and she's unmuted now, so I think she's ready. Adrian, please take it away.
Hi, everyone. As Connie mentioned, my name is Adrian Shaleen. I am the clinical research specialist with Angioma Alliance. I just wanted to touch on two quick things tonight. First is in regards to the CCM Health Index. So when this project begins, you will receive an email from me. It will probably come from coordinator at angiomalliance.org. That will explain exactly what you need to do to sign up for the project if you are interested. Once you are participating, you will be receiving emails from REDCap, which is the system that essentially is storing all of the data and the surveys that you will take. Once you click the link, you will be asked to agree to participate, and then you'll provide your email address, which will allow us a unique study ID to be assigned to you. And then once you start taking the surveys, if for some reason you need to come back later, take a break, you can pause and save your answers. So that's also a nice option. Once you've completed the first survey, you will be asked to complete 2 follow-up surveys. One will be at 6 months and one will be at 12 months. If you don't complete it, you will get reminder emails to complete it. And also just be aware that someone from the research team may specifically reach out to you to complete it as well if you don't complete it after the first few reminders. Outside of that, the other thing I wanted to touch on is just about Angioma Alliance registry. So if you ever have questions about the surveys, or you're having trouble signing up or getting back into your account, I can help you reset your password or help you navigate the website or the registry itself. I may reach out to you from time to time if there are new surveys that become available, if there's a survey that's in your profile that you need to take, because we really depend on those answers, especially once you've created an account. We really depend on those follow-up answers so that we know how you're doing as time goes on. And then the last thing is I may reach out to you specifically if I need to clarify an answer that you have already provided. Sometimes we start doing a project and we get into the data and there's something that might possibly be unclear. So we might reach out to you at that time just to clarify or provide some other information. And then lastly, I do reach out also if there is a new research study that has become available that you might possibly be eligible for, or I will reach out just to let the community know about new research that has come out and new data that we think is important for everyone to know. So just wanted to make everyone aware of when you might possibly be hearing from me, but also to let you know that I am here for you. If you ever have any questions about the registry, you can reach out to me. My email address is on the Angioma Alliance website if you need it. So thank you.
And I just wanted to do a quick little summary about the CCMHI. The recruitment is going to start as soon as the IRB at the University of Rochester says it's okay for us to start, which we're hoping is in the next week or so. If you are in the patient registry, you will definitely receive a notification that you are eligible to participate. Please, we need 400 people. So at least 400 people to do this, which means we need you. We have right now only 49 participants on this webinar, but we're hoping a lot of people will be watching it after the fact on YouTube. For this particular survey, though, because it is so important that we get a lot of people taking it, you'll also see posts on social media. And you may see it if you're receiving the email blast that I send out once a week. We are wanting to make sure that this is known far and wide. And again, what you do when you sign up, what you are signing up for is to take a survey now, to take the same survey-ish in 6 months, and then to do it one more time at 12 months. So it is really, really important that you are committing to all 3 administrations. If, for some reason you become ill or something, that's fine. But if it's just, I don't feel like it, that's not a good excuse. You got to do it, because otherwise it doesn't make any sense for you to be in the project. So commit to all 3. And then hopefully by the time we have that information, we will be able to take that to the FDA. And when FDA qualifies this as a valid tool, it can be used then in clinical drug trials as a measure. Maybe not the primary measure, maybe a secondary measure, but it can be used to show whether or not a medicine is having an effect. And that is what we all want. So your participation is critical. All right. The next thing is, Lisa, would you like to turn on your video and turn on your audio? You're promoted now. I know you have a big little thing to share tonight.
Hello. So I was here with you all maybe a month ago or so and promised that I would come back as soon as we had any additional information to share about the upcoming Sycamore study, which is the first industry-sponsored clinical trial in cerebral cavernous malformation. And the information that I have to share with you tonight is that the trial listing has gone up in clinicaltrials.gov. So the study is there now. It has an estimated study start date listed. So I'm not announcing that the study has opened. It has not opened yet. No sites are open yet. We have not enrolled any subjects into the study yet. No patients are participating, but the trial listing is live and there's a little more information about the study there.
And that, go to clinicaltrials.gov. I will send the link out. I can probably do that while we're answering questions. But one of the most important things there are the inclusion and exclusion criteria. So you will be able to see whether, just based on that, whether you think you might be qualified, might be somebody who could participate in the trial. In the end, it's the doctor that you're talking to who will make that decision. But, you can at least get a sense of, am I ruled out for something that would make me ineligible? And then you can continue on from there. So we're really excited to have that next step. And I think the other thing that I want to make sure that folks understand, this is different from the atorvastatin trial in that this is an industry trial, which means that information comes out differently. There are contracts involved. There's honestly money involved. There are shareholders involved. And so this process, I'm just learning, but please just follow along. Probably every webinar, we're going to have another new little snippet for you of some piece of information. Or again, and someone just asked, what's the name of the industry trial again? The trial is, Lisa?
The trial is the Sycamore trial. And Sycamore is the short name of the trial. The full name of the trial is a phase 2 randomized double-blind, which means no one knows who's taking drug and who's taking placebo, placebo-controlled clinical trial to evaluate the safety, efficacy, and pharmacokinetics of our drug, REC 994, in the treatment of symptomatic cerebral cavernous malformation. So a lot of information contained in the title alone. And if people have questions about the inclusion exclusion criteria that are listed on the site, we're happy to field those questions.
Okay. All right. We need your questions now. Q&A. I'm not seeing anything lit up there. Someone asked a question in the chat. If you could use Q&A, that would be really helpful. Were we extremely clear? Because I'm glad that we were. Barney is now joining us too with video. So if you have a question, this would be a good time to ask it.
One thing I'll offer while waiting for those questions to come in is, as Spencer described, the CCM High and the survey in order to further validate the study, the longitudinal version of this. We have also included the CCM High in the clinical trial that Lisa described and that is currently posted on the clinicaltrials.gov website. So the advantage of that is you have one portal of information that Spencer and his group will be looking at and collecting over the course of time. And then we will be doing the first assessment of how that instrument responds within a clinical trial setting. Again, this won't be definitive yet, but it certainly will be important in generating information to support the information as we move forward to getting approval for the instrument, and then having the instrument available for the community, not just for Recursion, but certainly we can contribute to the development of the instrument from that standpoint.
Thanks, Barney. And then I think a question that no one has asked yet, but I'm going to, is the CCM HI, is only Recursion going to be allowed to use that?
Currently, it is open for use, and we'll have people can reach out to my PI, Dr. Chad Hewall, to discuss the terms of that.
Okay. All right. Thanks. So it will be available to others then. All right. Someone in our Q&A queue asks, is REC 994 a drug used for other conditions besides CCM, or is it a new drug altogether?
It's a drug that's been used topically. So it's never been used as a medication that you take by mouth that, is acting across your entire body, but it has been used in a topical formulation in the past.
Okay. All right. So someone asked, is the Sycamore trial open for international participation?
Not right now. We're not doing any studies internationally for CCM right now.
Okay. And again, this is a phase 2 trial, and what you will see when you look at the clinicaltrials.gov listing is that they're enrolling 60 people only. So I think that is probably a number that they can hit within the confines of the US. I think trials go international when that becomes the number that they need become difficult to fill when they're in the US. And you guys can always correct me if I'm wrong.
Yeah, I would just say about that this is a first step. We have to collect some additional data with CCM patients, make sure that this drug, potential drug will be safe, and get some idea about whether it's going to be of benefit for patients. And then if all of that turns out to be true, we would move into a larger study.
All right. How was REC 994 identified as a candidate? What type of screening led to its discovery? Oh, you missed the last webinar, didn't you? But you can go on, Lisa, just summarize.
That's a great question. And I will give kind of a simple answer. And Barney, if you want to add anything to my answer that you think people would like to hear about, feel free. So Recursion's entire basis for discovering new drugs is that we take cells from humans and we put them in a petri dish, kind of like Connie was talking about, and we perturb them in different ways. So that might be by giving the cells a genetic mutation or exposing them to a chemical. And in the case of REC 994, we introduced this genetic change in the cells to make them like cells from a CCM patient, and saw that when you do that, you you get a certain change in how the cells look on the dish. So we take lots of pictures of these cells after we kind of shake them up with whatever our experiment is. And if you give Rec 994 to cells, then you get changes in the cells that sort of look the opposite in some ways of the changes that you might get if you give them a CCM genetic mutation. So we have a very fancy supercomputer recursion and lots of algorithms that go through this very large number of images that we generate and these cell culture experiments that we do, and tell us, oh, there's an interesting result here. There might be a relationship between the changes that you're seeing with this chemical and the changes that you're seeing with these genetic changes. And that's kind of how our platform matches up a chemical with a particular disease state. But then we go on to do experiments to confirm that there's a likelihood of benefit in giving this molecule to patients who have a particular mutation. So we have animal models of cerebral cavernous malformation that we have tested Rec 994 in, and putting all of that data together and understanding that it looks like a pretty safe molecule. That's how we made the decision to advance it as a clinical candidate.
Barney, you're muted.
And I can't get off my mute button. So as Lisa described, that's a preclinical process to get the candidate identified and then confirmed, at least in the models that we have, including the animal model. And then from there, we tested the drug in normal humans. And the point of that set of experiments is to try different dose levels and look at the safety profile to see how well it's tolerated. And so we did that in a format that looks at a single dose. So a subject would come in, literally receive a single dose, and then we look at blood levels and follow them. And then the second format is in a study in which multiple doses are administered to that individual over a period of time. And again, we're looking to see how well the drug is tolerated, as well as to assess the blood levels. And both of those studies look very good in terms of the safety profile of the drug. So based on the information that Lisa provided to you, and then based on the information from these human clinical trials, we feel comfortable now moving into a patient population.
Thanks. Did you, Lisa, there is a question in the chat that I'm not sure, I got distracted. Did you answer?
I didn't, but it's a great question about what.
Let me read it first.
Okay, sure.
How did Rec 994 change cellular morphology? Were the culture experiments done on endothelial cells?
Yeah, so I'm going to answer this backwards. The culture experiments were done on endothelial cells. So nice cell type for looking at a disease like CCM. The morphological changes that we capture on the system that we have, and I should mention that the system we have, which has grown a lot in the last 8 years, was built up around CCM as the first indication. So CCM is the disease that inspired the formation of this approach to discovering new drug candidates. And the way that we capture information about morphological changes in the cell cultures is high dimensional. So that's why we're using algorithms to analyze the changes that we see. We do what's called cell painting. So we go through with a number of different dyes and look at different tiny parts of the cell and capture very nuanced changes. So look at a large constellation of those nuanced changes. And from all of that data together, detect what we characterize as orthogonal relationships, meaning just that, oh, things seem like they kind of went east when we had this genetic change, and they look like they're skewing back west when we add this compound. So I know that's not a great answer, but there's not a simple answer to that question because we look at a lot of variables at one time when we do these image capture experiments.
Okay, thanks. Someone else was asking about spinal lesions. You guys, the phase 2 is brain and spine or just brain?
Brain, but that does include the brain stem. Does not include spine.
Okay, so that was the person has a thoracic lesion at T11, so that would not be in this trial. And to that person, I will be trying really hard to get more spinal stuff into the webinars, and I apologize that this and the last few have not included anything. So we'll try to make a change to that. I know
And
that
I'll say we'll try to get to you as soon as we possibly can
Yep, yeah.
in terms of generating data so we can head there.
And if
Okay.
there are people who have many lesions and some are in the spinal cord, but they also have a cerebral lesion that would qualify them to participate in the study, potentially those people will be in the study. If we learn more about those lesions over the course of the trial, or we learn more about how Rec 994 impacts those lesions, potentially that would inform another trial.
And I can also share, not specifics, but I can say that we have a scientific meeting coming in 2 weeks. That is our annual international scientific meeting held virtually this year. But it does include new information about spinal CCM. So there's more to come. It will have to wait until it's published before and peer reviewed and published before it can be shared with the community. But there's definitely a lot of work, and honestly, a lot of new work and new interest in spinal lesions. Okay, somewhat same person asked, in theory, is it hoped that Rec 994 could correct or cure existing lesions or prevent formation of new lesions, or both?
I would say that if we look at where we are today, our expectation is that the drug will prevent the development of progression. We would hope that it would also potentially reduce the amount of iron that is deposited around the blood vessels. So if you know you have the blood vessel and it leaks blood, well, there's a deposit of iron there, and that iron is related to the generation of seizures and headaches and so forth. So if we're able to reverse some of that, then that would also improve how you feel. At this point, I'm not going to say that we're going to completely reverse. Lesions are already there. At the baseline, our expectation is that we can at least keep things from progressing.
I would add to that this study that will be opening, in the near future is primarily a safety study. So we're looking to understand, when people take this drug candidate, is it safe? Are they feeling okay? Is it causing them to have any side effects? And we appreciate everyone's patience, because this is very much a stepwise process. We'll be learning more about how to study CCM over the course of this trial. Now, obviously, if we were to have some finding in the study that wowed everyone and knocked our socks off, we would follow up on that. But it will be a long process to understand what we can hope to expect using this molecule.
We, as I mentioned, that we had done the studies in normal humans to look at safety and blood levels, but this will be the first time we've gone into patients, and we really need to ask essentially that same set of questions in a group of patients to make sure that the drug is handled the same way. We expect that it will be. There won't be a difference between a healthy, normal person versus someone with CCM, but we need to verify that. So that's part of what we're talking about here. We have built in some assessments of potential efficacy. So for example, the MRI. So we build in some things from that standpoint, but an important piece of this is the CCM high, in the sense of trying to see how individual symptoms might change over the duration of the study. So it is really designed as a safety, tolerability study and pharmacokinetic study. So we check these blood levels, but at the same time, we are going to be looking at first indications of efficacy in the course of the study as well.
Yeah. And so I think what Barney was trying to say is they want to know if you feel better. That's part of this, is making the CCM HI, making sure that it is a tool that can be used to determine whether or not somebody feels better with CCM, specifically someone with CCM feels better. All right. All of a sudden, we're getting a flood of questions in. Is it known if Rec 994 crosses the blood-brain barrier when administered systemically?
Animal data suggests the answer is yes, but we have to prove that in man. So can't give you a definitive answer yet.
All right. And then the same person is asking, it was approved as a topical. Is there any information about administration as a transdermal?
It's not been approved as a topical. Lisa mentioned that it's been tested, but it hasn't been approved as a topical drug, and it's being used to treat radiation burns for people who've had radiation therapy. That's the reason it's being tested, but it's still in the testing phase for that indication as well. And to my knowledge, no one has looked at it as a transdermal. That doesn't mean no one has, at least to my knowledge, that no one has.
Someone is asking, where will we have to travel for the Rec 994 study?
Stay
Everyone
tuned.
has to go to Bermuda. No,
We would like
Lisa can answer.
with a nice travel stipend that will buy me umbrella drinks.
Yeah, I will just say about that you'll have to stay tuned so that as we have sites opening, we can tell you where they are. But it is our goal for the study to be accessible, within those first 60 people who are able to join the study. So it's not like we'll have everyone traveling to one location in the country. We'll try to have sites spread out so that people can travel a reasonable distance to participate.
So the answer is they're working on contracts, and when they have the contracts in hand, signed and everything, then they can make announcements. Until then, we just trust that it's going to be near you. Someone else has asked, do you foresee any significant health risks for CCM patients once the trial drug is introduced into their system? Any chance it could adversely affect existing lesions?
Well, to answer the last part, is there any chance? It's always the challenge when you're evaluating any new drug in any indication for that matter. Is there risk? As best we can tell right now, again, I can't prove that. I can't tell you for sure. But based on the data that we've collected so far, we think the risk, if there is any, is minimal. So, but that's, as Connie pointed out earlier, that's why you do clinical trials like this. You have to be checking constantly to, on one hand, is the drug safe? Is it tolerable in this patient population? We can give it to a whole bunch of normals, but that doesn't necessarily tell us what's going to happen with the patient. So is it safe? And then does it work? And does it work in a way that's sufficient to show benefit for the individuals taking the drug? And those are the pieces we have to continue to evaluate over a period of time. And unfortunately for us, for the whole world, there is no single study that's going to answer both of those questions in a very short period of time. So it will take time to get the initial safety readout, and then based on those data, it'll take longer to get the efficacy readout as well.
And I would just add to that, we applied this molecule in mice that had genetic mutations in the CCM genes, and we did not see any worsening of the lesions in those mice. That said, mice are not tiny humans, which is why, as Barney just stated, we have to do an experiment in people.
I was trying to answer someone offline.
Yes.
Great. If phase 2 is deemed a success, what would phase 3 entail?
Bigger study. So yes, if phase 2 is deemed a success, then what we have to do is to look at the data. Keep in mind, as Lisa pointed out a moment ago, first safety, then we move to efficacy. So if we have safety, then we'll feel more comfortable expanding the number of patients and expanding the geography. So somebody did ask about an international trial previously. If we can hone in, if it turns out that the data from the CCM high indicate that that instrument is responsive in terms of how people feel, that actually becomes a major help in terms of trying to look at efficacy. So we'd have that, which helps on the symptoms. Do you feel better? Has it made a change in your life? And then some of the other things like looking at MRIs, we can match that information together and say, yes, this drug actually does have benefit. So we would most likely expand the phase 2, larger groups, confirm the efficacy. And depending, we might be able to roll that into a phase 3, larger confirmatory trial. Maybe it's a separate trial that's subject to negotiation with the FDA or the European Medicines Association or agency to try to make sure we've got the best, most efficient development pathway. But given that this is a rare disease, we're not looking at really large trials to do this. We're banking on important data being generated in relatively small trials.
And our stance at Recursion is that we would like to deliver anything that we find that might be of benefit to patients. We would like to deliver it to as many patients as possible, as quickly as possible. So I just want to underline that our team works very hard to design studies that will move potentially beneficial trials to patients as quickly as possible.
And Rec 994 has orphan drug designation, right? So there is already an understanding at FDA that this is a rare disease, and the expectations for the trial are therefore somewhat different than it would be for high cholesterol.
Yes, yeah, absolutely.
I think I am, let me double check here, but I think we are pretty much done with time and done with questions. So that worked out well. Any final words from anyone before we close out for the night? No? Okay.
Thank you for inviting us to join you. Always a pleasure.
You're going to come back again once you have sites to tell us about and enrollment information. And also immediately following this, the support group is going to be meeting. In the chat, there was a link to register for support group. It was up a couple of times, so I'm hoping you had a chance to click on it and get yourself registered, and the group will be probably very chatty. They usually are after the webinars. Thank you all for coming. Thank you, Spencer. Thanks, Adrian. Thanks, of course, Barney and Lisa for being part of this evening. Thanks and good night.
Thanks very much for everyone.
Thank you.
Thank you.